Hello and welcome to Neuro3301. It's time once again for our course. It's the beginning of another week, my friends, and here we are yet again learning about different things. Today's topic is neurodegenerative diseases or diseases. Perhaps all of our brains have degenerated a little bit since the start of the semester, since the start of the pandemic. It's just the way she goes. But the neurodegenerative conditions we'll talk about today are not the same as the sort of normal brain worms that we tend to get around this time of year. They're a different sort of a thing. So we've got a lot to cover today, folks. Let's just jump right into it and begin the lecture. So yes, today's topic is neurodegenerative disease. And we're going to go over basically Alzheimer's. That's the main one that we're really going to get into today. The symptomology of it, some of the genetics of it. Then once again, you'll have to endure a discussion of linkage in genetics, but it'll be our last one. And then we'll go into a little bit about the epigenetics and various confounds having to do with cell composition. And that'll be it. So you'll hear a little bit about the genetics and epigenetics of neurodegenerative diseases. So Alzheimer's disease is sort of the classic or the most common type of neurodegenerative disease. It's not the only one. If we had a lot of time to spare, we could get into other ones like Parkinson's or Pick's disease and various other kinds of things. However, Alzheimer's is fairly well studied in the world of genetics and epigenetics, so I think it makes a pretty good test case. The primary symptoms, of course, are loss of previous memories, as you would say, retrograde amnesia, as well as deficits in storing new memories. This would be anterograde amnesia. People with Alzheimer's disease also have deficits in executive function. By this we mean the ability to execute complicated tasks with multiple steps. And here this is coming from failures in the short-term memory as well as a sort of a breakdown or a dysfunction in the frontal lobe. I always think that when you're looking at the gradual development of Alzheimer's-like symptoms in the elderly, one of the things that really starts to go fairly early on is cooking. Cooking, if you think about it, is a fairly complicated task. You require, to cook well, you need a lot of planning, the ability to sequence multiple different events, keep track of things as they're on the stove, time things out, plan things in the right order. And when you start to see problems in the kitchen with people who have had, cooking experience their entire lives, that's a good sign that things are not all well in the brain department. Other symptoms of Alzheimer's would include agnosia, the failure to recognize objects, apraxia, impaired motor function, and aphasia, which is impaired language function. So quite a multifaceted disease, really, that sort of sweeps through the brain and takes out a lot of different levels of functionality as we go. Now, Alzheimer's is a progressive disorder. It begins in a very subtle way and will progress gradually toward higher and higher levels of impairment. During the early and later stages, the deterioration is quite, comparatively slow, but during the middle stage, it can become quite rapid. So that ultimately, after you've been diagnosed, the average the average remaining survival time you have is about 8 years. The symptoms really tend to appear as mild absent-mindedness at first. If you analyze, people have looked at this, and if you analyze the written and spoken language of people that's been transcribed, you will see that the language becomes simpler and more concrete. By concrete, I mean it uses fewer analogies, fewer bits of alliteration and clever prose, I guess. It just becomes more straightforward. Sort of the way that little kids write, it just becomes simpler. And this is the kind of thing that you can analyze in the text just based on, kind of patterns of word usage. People will do this actually it's probably not a great thing to be doing, but people will do this with public figures. If public figures are known to have a lot of a long history of writing or speaking, you can analyze their patterns of speech and say, they've gotten a lot simpler in recent years. Maybe they've got some kind of hidden dementia that nobody's talking about. As the disease progresses, the symptoms become more and more severe. The language problems continue to, or the language skills continue to decline. Noticeable deficits in long-term memory begin to appear, and you also start to see problems with affect and emotion. Individuals could become irritable or aggressive, or prone to emotional outbursts, either hysterical sort of laughing or anger at various inappropriate times. Individuals can also become confused and wander away. Now, the progression through this neurodegenerative disorder can be scored by histological staining. So when you're looking at the brain, using, say, staining for the tau protein, which indicates neurofibrillary tangles. Various other markers contribute to a system called the BRAC score. This is made up by some guy named Brack, and it's basically just a way of quantifying the degree of histological neurodegeneration. So the higher the Brack stage, the more severe the neurodegeneration is. And you'll see this when we look at some of the epigenetic studies. If you want to understand, if you want to link this or that, like link any kind of epigenetic or tissue marker to Alzheimer's disease, you need some kind of objective way of scoring how advanced the disease actually is. And the Brack stage tends to be what they use for that. So we'll learn more about the Brack stage toward the end of this lecture. Now, this ultimately results in the individual becoming quite dependent on caregivers, oftentimes institutionalized. Extreme apathy, exhaustion are common at this stage, and muscle mass may deteriorate substantially. Language skills often become non-existent at this stage. Some emotional signals and words are still understood, however. So it's a terminal condition that that sort of inevitably results in a terminal decline toward complete dependence on caregivers and complete disability. The rate of the decline and the age of onset, however, does have some level of variability. Alzheimer's typically appears during the 60s and 70s, though early onset Alzheimer's can appear as early as the 30s. This would be called pre-senile dementia, senile meaning old. Whenever you see that prefix in language, it just means old, sen. So like Senate, for example, is a place where old people are. Believe it or not, that's the literal meaning of it. Senior, same thing. Early onset Alzheimer's disease tends to progress rapidly and viciously. And so if you begin to develop Alzheimer's in your 30s, you're not going to have a good time. That is a very, very rapid course of disease that you'll be experiencing. And those early onset cases tend to have a much higher genetic contribution than the late onset type. Alzheimer's accounts for about 50% of the cases of dementia, and in the US at least, it affects more than 5 million Americans. So we're sort of justified in focusing on this if we're trying to understand neurodegeneration and dementia, because this is, pretty much the most common single type. Late onset Alzheimer's disease is genetically distinct from early onset. The early onset is actually kind of an interesting case. It comes in sporadic and familial forms. Familial forms meaning ones that you can look at the pedigree and see a pattern of inheritance, and sporadic meaning that it's genetic, and it's putatively genetic and comes on early, like the familial form, but there's no family history of it. So you might think it's due to a de novo mutation or something like that. The familial form is linked to mutations in genes such as the amyloid precursor protein, presenilin-1, and presenilin-2. These three genes account for the main disease-associated mutations for early onset or presenile Alzheimer's. The amyloid precursor protein, APP, is post-translationally processed into amyloid beta. We don't know, of course, what amyloid beta does, but it is the source of the amyloid beta plaques, which you see accumulating in the brains of individuals with Alzheimer's. Presenilin-1 and presenilin-2 code for the presenilin enzymes, and these regulate the cleavage of the amyloid precursor protein through gamma secretase. So these three genes that are highly associated with early onset familial Alzheimer's are all ones that have to do with either producing amyloid beta or processing the amyloid precursor protein into amyloid beta. And it's not surprising, I suppose, given this fact that much of the work on Alzheimer's drugs and treatments has focused on trying to reduce the levels of amyloid beta in the brain. I should say, this doesn't really end up working in practice. There have been many big, high-profile clinical trials that have resulted in essentially no improvement in Alzheimer's conditions, despite giving people antibodies against beta amyloid, despite giving them various kinds of treatments that, in mouse models, do actually lower the levels of amyloid beta in the brain, still apparently do nothing to humans. So there's this clear link between amyloid beta and the disease that we can see both in the tissue histology and in the genetics of early onset Alzheimer's. Yet it doesn't seem to be as simple as just getting rid of beta amyloid to cure Alzheimer's. Now, when we get into late onset Alzheimer's, which is going to be the main focus of this class, there is also a genetic cause there. Twin studies seem to suggest there's about a 40% concordance rate, which is relatively low as far as these kinds of things go. With these severe conditions that, oftentimes, that sort of uniformly result in severe impairment, I think oftentimes you'd expect a higher genetic contribution. But the twin studies say it's about 40%, and that means that the better part of the risk of Alzheimer's is actually environmental. Whether it's shared or a unique environment is a separate question. But it does say that over your entire life, the sum total of your experiences up to your 60th or 70th year, all of that goes into sort of modulating your risk of Alzheimer's disease. And I guess this does make you think about what it means for something to have an environmental or a non-genetic contribution when it's something like Alzheimer's that comes on in your senior age, 60s or 70s, versus something like, say, ADHD, which is typically diagnosed in very, very young children. The fact is that in young children, they've just been alive for less time. So if there's some kind of environmental factor that modulates or that affects disease risk, they'll have been exposed to it for less time. So think about that for a little bit and consider the complexities of trying to suss these things out when these conditions are diagnosed on such completely different timescales. The most well-characterized single Alzheimer's risk gene is apolipoprotein E. This is normally involved in cholesterol transport. But we'll get into a number of others as well. So with that introduction out of the way, let's leap into the world of GWAS. So in 2019, the International Genomics of Alzheimer's Project, the IGAP, published a meta-analysis of late-onset Alzheimer's disease that focused on over 21,000 cases and over 41,000 controls. So pretty good numbers, although we've seen studies that approach 100,000 cases and controls. So it's not quite as giant as it could be, but it's these GWAS studies just get bigger and bigger and bigger. Every time you do a new one, you can add the results to the old ones that you've done and build up larger and larger numbers. So let's take a look at the Manhattan plot and see what there is. Now, you're, I'm sure, used to seeing this kind of stuff, and my experience in trying to go over the results and teach GWAS studies has shown me that there's not a huge amount, oftentimes people don't get a whole lot out of just going through the genes one after the other. That's, in some sense, it's just sort of too complicated. If you look at the number of genes here, what CR1, BIN1, TREM2, CLU, many of these have fairly obscure functionality, and it's rather difficult to go into detail about what they all do. Although, of course, we see ApoE is still the top hit here over here on chromosome 19. So rather than get too into the weeds on what these genes do, what we can do instead is take a bigger picture look about what the GWAS is telling us. And I guess here I wanted to get into a discussion of linkage and maybe just a little bit more about how to interpret these figures. So what we have here, so let me zoom out a bit. This is called a Manhattan plot, and the reason it's called a Manhattan plot is because these points pile up on each other and look like towers in a city skyline, okay? And, Manhattan sort of has a famous skyline. Now, why is it that there is an index variant that is the most significantly associated? Remember, the higher it is on the y-axis, the more significant the association. Why is it that there's a highly significant SNP at the very top, but then a whole bunch of other SNPs that are, physically very close to that SNP with sort of progressively lower levels of statistical significance? What does that mean? Well, the top variant is called the index variant. It's also called the sentinel variant. It has a couple of different names. And then there's also a range of linked variants. The linked variants are significantly associated with the condition as well, but they're significantly associated by virtue of their linkage to the index variant. So let's go in for another dip into the old linkage discussion. As I've said a number of times now, linked alleles tend to stay together during meiosis. During the homologous recombination phase, the chromosomes kind of get together and swap out different parts. From different parts of the maternal and paternal chromosome are swapped together or swapped to create new combinations of chromosome fragments for the gametes and therefore for the offspring. This means that offspring are not getting just one-for-one duplicates of their parents' DNA. They're getting a novel mixture that incorporates both of their grandparents, essentially. And that means that you will end up with a mixture of traits. Your offspring will end up with a mixture of traits that don't exactly match yours or anybody else in your family. But what's important about this, that I think is a fairly straightforward fact, is that recombination happens between consistent breakpoints. So these fragments of DNA that exchange places during homologous recombination, they're not chosen randomly. There are actually specific spots along the length of each chromosome where the DNA will break apart and those chunks separate in the same place across different individuals. So these breakpoints are repeatable, they're measurable, and they're consistent between individuals. So that means that alleles actually associate with each other in a non-random way because these breakpoints are sort of, they're fixed, they're not particularly variable. This is a direct contradiction with Mendel's law of independent assortment, I should say, because it means that genes are not being inherited on a completely random basis. They are linked to one another to a certain extent. Now, as I've said a number of times, linkage is related to the physical distance along the length of the chromosome. So regions that are, statistically speaking, regions that are closer together tend to stick together during meiosis. The further away two different regions are, the less chance they are of being linked. And because of this, linkage can be used as a way of mapping genes to locations along the chromosome. Now, this is all, I think, stuff we've gone over a fair bit. It's the source of it's the source of linkage studies, for example. It's the source of our ability to link traits together in their heritability and so forth. In the genomics age, so I guess in the old-fashioned sort of pre-genomics age, finding out linkage between two different locations was fairly tricky, and it involved looking on the physical chromosome for evidence of linkage, as well as doing lots of fragment length genotyping and so on and so forth. So it was quite a complicated question. But as with many things, the genomics era has made this considerably more understandable. So this is where we introduce the term linkage disequilibrium, or LD. It's a term from population genetics referring to the non-random association of alleles at two or more locations in the genome. So it's named this sort of as an answer to the hypothetical state of equilibrium, which would be the assumption of Mendel's law of independent assortment. Every single allele that you could possibly have is inherited completely independent of one another and in a completely random way. That means that if you look at the allele frequency or the co-occurrence of alleles in an individual, they would be proportional to their frequency in the overall population. So that's what's proposed by the law of independent assortment, is that everything is basically just random and there's no linkage or connection between any of the different alleles in the genome. Disequilibrium is simply the reverse of this. Wherever there is linkage disequilibrium, it's where the apparent linkage of two different alleles is higher than you would expect on the basis of this hypothetical random state of assortment. So let me see if I can put this into more concrete terms. But first, I'll throw a media recommendation your way. This is from our friends at the Khan Academy. This is a video about gene mapping, and it's just 13 minutes long. And if you, I guess to me, this is the kind of thing that is potentially hard to understand. It's the kind of thing where you might listen to one person's explanation of it and have it continually frustrate you, but then somebody else might explain it a different way, and it could be a completely a much easier story to understand. Oh, so we've got a question in the chat here. So Nikita asks, I just wanted to clarify, if someone has a certain variant of ApoE, that indicates they're at risk for Alzheimer's disease. But if it's mutations in APP, PSEN1, and PSEN2, which are the actual mechanisms for the degeneration. No. So what we're talking about here is the difference between early onset and late onset Alzheimer's. Early onset Alzheimer's is the kind that you get typically when you're quite young, in your 30s and 40s, and that is almost, genetically speaking, it's quite separate from late onset. So the early onset Alzheimer's disease involves things like APP, PSEN1, and PSEN2. These are sort of point mutations that are inherited quite readily across families, and thus there's a family history for it. The late onset Alzheimer's, which makes up most of the Alzheimer's you actually see in the world when you go to nursing homes and when you look at old people kind of creaking around the shopping malls and whatnot. This is a separate condition that just happens to have the same name, more or less. And there, the number one most associated gene is ApoE. Despite this, and having a fairly non-overlapping set of genetics, early and late Alzheimer's disease have a similar phenotype in the brain. So they're both associated with beta amyloid plaques and neurofibrillary tangles. They both produce the same sort of symptoms, although typically early onset Alzheimer's progresses much more rapidly, and of course it's early onset. So what I'm saying here is that the two conditions manifest similarly in terms of the neuroscience, but the genetics are comparatively they don't really overlap a whole lot. The genetics for early onset are the APP, PSEN1, and PSEN2, and the genetics for late onset are the ones that I'm showing you that I showed you in the Manhattan plot earlier. Does that make sense? Okay, hopefully that makes sense. From here on out, we won't be talking about early onset Alzheimer's. I guess that would be another topic for another class. Any of these things you could spend class after class on studying in depth. I'm trying to give you kind of a surface level just overview of what the literature is showing and what we know so far, and then also how the work is being done. So yeah, I was just sort of saying that this would be the kind of thing you'd want to look at if you're having trouble understanding linkage. This gives you a bit more detail on how genes are mapped out and how linkage is calculated and established. So I think this is a nice thing to have in mind when you're doing your studying. Now, it's kind of funny to think that most of studying could be done just by watching YouTube videos. I know that when I was in your shoes, I would oftentimes find it was easier to read different books about things. Sometimes my professors just couldn't, the way they explained something just made no sense to me, or the way my textbook explained it made no sense to me. But I could read a different textbook or hear somebody else explain it, and sometimes it would be crystal clear. So there really is something to be said for teaching styles, I guess, and how well it reacts with certain types of learning styles. Maybe that can be a big data problem. Maybe we'll figure that out using machine learning. Anyway. So talking about linkage disequilibrium, the concrete example I wanted to give you goes like this. imagine you have two loci in the genome that have various alleles, but imagine you've got alleles B and S, okay? Both are present in the population with a frequency of 50%. So at this location, you know, little b is 50% and big B is 50%. And then at this location, same thing. Little s is 50%, big S is 50%. So a simple question is, for any given person, how often would you expect them to have both big B and and big S, or little b and little s? What would be the frequency of co-occurrence for these two alleles? Now, if the two locations have no linkage whatsoever, then the odds of seeing big B and big S combined would be equal to their allele frequencies multiplied against each other. So they're both present with a frequency of 50%. So your odds of having B, the big B, is 50%, and your odds of having big S are also 50%. So your odds of having both of them at the same time is just 50% times 50%, i.e. 25%. Now, that's the null hypothesis, or sort of the baseline assumption, if they're not linked. However, if they're linked, what you would see is that on the population level, you would see them co-occurring more often than 25%. So, for example, if you observed a 40% co-occurrence, these loci would be in linkage disequilibrium with each other. Linkage disequilibrium just means that their, equilibrium would mean that they're random and not connected whatsoever. So the chance of seeing them both together is just the allele frequencies of each multiplied together. But if you see something other than that, then they're in disequilibrium with each other, and they are therefore linked. So another way of putting this, I guess, is if you see co-occurrence rates of the two alleles close to what you expect by chance, 25%, that would indicate there's a high rate of recombination between the two markers. So this is another way of expressing linkage. We've talked about it in terms of centimorgans and recombination events per meiosis and these kinds of things. But the kind of advantage of studying LD is that all you have to do is know the allele frequencies for everything and then measure the observed allele frequencies. So the allele frequencies of the alleles on their own versus the co-occurrence of these observed in real life. And those two pieces of data on their own will be everything you need to know to calculate linkage disequilibrium. And that will let you know, or that will give you a very strong way of inferring how linked these really are on the chromosomes. So a common measure of linkage disequilibrium in genome-wide studies is R², which is really it's literally just a correlation coefficient, or it's the square of the correlation coefficient between the two markers. So if B and S are perfectly correlated, every time you have big B, you have big S, and every time you have little b, you have little s, and there's never a scenario where you have big B and little s, or the two don't match. That would be a perfect correlation, and it's rarely ever going to be perfect, but typically a high correlation is very strong evidence that these two locations are linked, and a low correlation means that there's no linkage. So we can take this concept of correlating the frequency of different alleles along a chromosome and kind of play this out to its logical conclusion. You can take every different SNP and correlate it with every other SNP, and you'll end up with this kind of thing. Yes, so Ali asks, if R² is 0, then there's no LD. That's correct. If R² is 0 or close to 0, then it means that the two alleles are being inherited independently from each other. If the two alleles are being inherited together, then they're going to be correlated. But if they're inherited separately from each other because they're not linked, then there's going to be no correlation between the two. So you can go and measure the correlation between allele frequencies across the entire genome, and you end up with these sort of correlograms. Now, I think I showed you a correlogram in, what lecture was that? Might have been schizophrenia. It was one of the ones where I was showing you the genetic correlation between a range of different phenotypes. And I showed how you could start with one phenotype and then look over and see its correlation with another phenotype. And it was all kind of graphed out in a big block format. This is also possible in the genome, and you end up with these things here. So what's happening here is the algorithm essentially is just starting at the first SNP, and it's measuring the correlation between the first SNP and the second SNP, and that's given by the number in this cell. So the R squared is 0.96, or the correlation is, the R squared is, R squared times 100 is 96. I don't know why they do it that way. I just to get rid of the decimal point. So that's a very, very strong correlation. And then the correlation between the second SNP and the third SNP, they're very close together, is also 96. And then the correlation between the first SNP and the third SNP is 92. Those are all very, very strong correlations. However, the correlation between the third SNP and the fourth SNP is 4. That's very, very low. So these three locations are linked to each other because they have very high correlation values, but they're not linked to the next one down. The same thing is happening over here on block 2. These SNPs in this block are highly correlated with each other, but they're not very well correlated with anything downstream. And you have another correlation here in block 3, block 4, and block 5. So these blocks of intercorrelated SNPs, or SNPs that are in high linkage disequilibrium with each other, are called haplotype blocks. So let me just erase some of my little markups here, because I have something else that I wanted to bring up about this. Okay, back to the pen. So you can look within blocks and see that the locations there have high levels of linkage with each other, and you can see that between blocks there's not a great deal of correlation. So you can see that all this kind of uncorrelated area here means that your status for this allele, for example, well, let's just take the first one. So the first one, the first SNP and the last SNP, you can find their correlation by looking at the very end of this triangle, and that's a 3. So that's not very well correlated. And you can kind of go down the line. You can see that your status for this haplotype block, whatever SNPs you have here, has no relationship whatsoever with your status for the other haplotype blocks. They're inherited as discrete units. So within the block, these variants all tend to hang together. So if you know one allele, you know with a very high level of confidence what the other alleles are going to be. But then between blocks, there's very little relationship at all. Now, this is a fairly important aspect of heritability, because it shows in a sort of practical way how linked variants are inherited together in these blocks. It was a fairly early project in the genomics era, around 2003. The project was called the International HapMap Project, which aimed to characterize the linkage disequilibrium for the full range of the human genome. This is fairly important for a reason that I'll get into, I think, in the next slide. The reason is that LD scores and haplotype blocks allow you to impute missing genetic data. And the reason is this. If you know the haplotype structure of the genome, it saves you a lot of trouble when it comes to genotyping. Because, well, consider this. Most sequence doesn't vary at all between people. If you're put yourself in the position of somebody trying to design a GWAS study. You know that the human genome is almost 3 billion bases. Now, one thing you could do is just sequence the entire thing. That would be perfect. If you could do that for everybody, for hundreds of thousands of people, you would have all of the power in the world to make your determinations. That's not realistic, though. You have to genotype people at a smaller handful of things. So the big question is, where do you genotype? Where do you measure people's genotype in order to get the most information about their situation? Well, the first thing you have to know is that there's only about 1 SNP per 1 kb of genome. So for every 1,000 base pairs, on average, there's about 1 SNP. And there's actually fewer in the exomes. There's only about 1 every 2 kb in exomes. So it would be pointless to study anything that's not a SNP. Obviously, if something doesn't vary between people, there's no point in looking at it as a potential cause of disease. At least, well, strictly speaking, there is a point, because it could be a very rare variant. But if in the general population it doesn't tend to be any different between people, then there's no point in looking at it as a potential cause of a common condition. Now, if you take some time before you get started with your GWAS studies and just look at the whole range of human genetic variation by sequencing tons of genomes, you can build haplotype blocks or haplotypes of SNP alleles that frequently co-occur. The reason is that, SNPs are linked. You don't need to, suppose you're talking in this scenario and you're looking at haplotype block 2. Well, the first SNP is very highly correlated with all the other SNPs in the block. So really, you don't need to know all these different SNPs that are in this block. You just need to know the first one, because the first one is so highly correlated with the other ones that it makes measuring the other ones kind of redundant. And this is what we're talking about here. By building up a genome-wide map of haplotypes, you can figure out what parts of the genome are distinguished haplotypes and which parts are actually worth measuring. That means you only need a few tag SNPs to identify an individual's haplotype, and from that you can impute all of the other alleles that the person has. So here's an example of what this HapMap project was really trying to do. These are the reference haplotypes, and these show all of the SNPs in a given haplotype block. So TC, TA, TT, GC, etc. These are all variants that could exist at these different positions within a haplotype block. And a haplotype block, I should say, could be quite big. It could be tens of kilobases if you wanted it. It can be quite large physically on the chromosome. So by sequencing a whole bunch of people, you can build up these reference haplotypes and say, okay, well, wherever there's an A in this position, there's almost certainly going to be a T and then another T right after it. And wherever there's a G in this position, it's going to be followed by 2 T's. And wherever there's a C in this position, it's likely to be followed by 2 A's or maybe an A and a C. You can build up these reference haplotypes. And then maybe over here, if you ever have a T here, it's typically followed by 2 C's instead of 2 T's. You can build up these reference haplotypes and also their frequency in the population. And then when you go to do your genotyping for your study, you don't need to genotype every single SNP in this haplotype block. You just need to genotype a selection of them, like A, G, C, and you can guess on the basis of the linkage disequilibrium and the correlation between these SNPs what the other ones are going to be. So you can fill in the missing values, and where you're not sure, you can just put 2 of them on top of each other, and you can say you have a lower confidence in that particular imputation. You see AC is here because the person could either have an A or a C in that position. So that's the aim of doing a haplotype construction, is by studying the linkage structure of the genome, it saves you a great deal of trouble when you're doing your genotyping, because you can get a lot more information out of the same amount of data. Out of a smaller amount of data, I should say. So here's a schematic of an LD map, and I've given you a handful of SNPs here, and we have 2 haplotype blocks. One haplotype block only consists of 2 SNPs, of course, and the other one consists of 6 SNPs. So they're right here. The correlation, or the R squared, is given by the color scale. So dark red means that these SNPs are very highly correlated with one another. That is to say, they're in high levels of LD. And white means very low correlation. So this is just the exact same as the plot I was showing you before. These 6 SNPs are in high LD with one another. And these 2 SNPs are in high LD with one another. But then these 2 blocks are not connected at all. They have no LD with each other. So we've got 8 SNPs altogether here. Now, if you want to save time, you wouldn't really need to genotype all 8. Because these are so highly correlated with each other, it would be sort of redundant. Instead, what you could do is just genotype this one and this one. So you could genotype SNP 3 and genotype SNP 8. And because there's such a high level of correlation, that will tell you with a fairly high degree of confidence what the genotype is going to be for the other SNPs in that block. Do you see what I'm saying? So it's a way of saving a lot of effort and resources in your genotyping effort, or in your genotyping operation, by leveraging existing knowledge of human genetic variation and the sort of the linkage structure of the genome. So hopefully that made sense to everybody. I realize it is just sort of a strange, kind of sort of obscure concept. I mean, hopefully it makes sense, but it wouldn't be totally surprising if it was a little tough to understand because it is it's just a strange thing, I guess, to think that this is the way the genetics work, but it's true. So what does this mean? I digressed a great deal, but we were trying to get at something about this Manhattan plot. The reason you see one SNP on top of a pile of other SNPs is because those SNPs below the top one are in LD with this index variant, okay? And if they're in LD, it means they're part of the same haplotype block. And if they're part of the same haplotype block, it means they're, number one, very close to the index SNP, and number two, well, I guess maybe number one is the most important thing. They're very close to the index SNP, and because they're, I guess number two is they're inherited together, and thus they would have almost the same level of association with the trait as the index SNP, because they're sort of synonymous, in a sense, they're sort of synonymous with the index SNP. They're inherited together, they're very nearby. You may be less confident in them because you're imputing them, not measuring them directly. That's probably why the p-value is lower. But fundamentally, they're a part of the same family, and they're located in the same region. They might have been measured or they might have been imputed based on haplotype. So this is important to consider because the index SNPs that we focus on in our GWAS studies are chosen by the designers of these microarrays to be representative and accurately genotype the haplotype block. But that doesn't necessarily mean that they are the true causal SNP in a given condition. It could be that the SNP that's really actually causing the problem is one of the ones that it's linked to, not the one itself. And this matters a fair bit because, as you might recall from our lecture on functional genomics, things like transcription factor binding sites are quite relevant to understanding why it is that a single nucleotide variant could have such a large effect on molecular biology. Now, the transcription factor binding site is determined by the sequence of the DNA. So it really matters where you look. If you look at the exact place of the index SNP, you might get the wrong impression because it might just be that the index SNP itself is in linkage with the real causal SNP that might be somewhere else nearby. So you have to know where to look, and that becomes quite challenging because the way genotyping works, you only genotype a handful of places and then impute everything by the LD scores. So it does mean that the question of where the exact causal variants are located is not exactly given by the GWAS method. The GWAS method just points to a rough area. So in a way, it's kind of like, it has a similar sort of problem to those old-fashioned linkage studies that were very imprecise. They could probably narrow you down to within a few hundred kilobases of where the action was, but no further than that. And GWAS studies, of course, are much more accurate than this, but still, there's a little bit of uncertainty as to where exactly the truly causal variant is located when all of these nearby variants are in LD with one another. So that's why a Manhattan plot looks the way it does. That's why a Manhattan plot is tall and has a skyscraper-y kind of appearance to it. Let's move into epigenetics now. As always, our GWAS studies point to brain-related genes and genes involved in various aspects of cell physiology and apoptosis and neurodegeneration and so on and so forth. But they don't explain everything, and especially for a condition like Alzheimer's, where the environmental contribution appears to be rather high, we have to look somewhere else to understand the rest of it. So the epigenetics of Alzheimer's has been studied reasonably well, and I'm going to present to you a case study coming out of the year 2014 that sort of highlights some of the technical challenges in this field and gives you a sense of where the epigenetics is headed and the challenges that we have in doing this kind of work. It's kind of an example, I suppose, of this common phenomenon in science where two studies are done separately by, separate groups of people, yet look at the same thing and arrive at very similar conclusions. So the first article, they were both published in Nature Neuroscience. They actually do have a fairly high overlap of authors. John Mill is in both of them, for example. But the first one says, Alzheimer's disease, early alterations in brain DNA methylation at ANC1, BIN1, RHB, DF2, and other loci. And then the other article says methylomic profiling implicates cortical deregulation of ANC1 in Alzheimer's disease. So they're both kind of agreeing that this methylation levels at ANC1 in the brain have something to do with Alzheimer's disease. So we could look at both of these, but we don't have time to look at them both. So I chose arbitrarily to focus on Katie Lunnon's paper. This is the one here. What they were using was a method for studying genome-wide methylation called the Illumina 450K microarray. This gives you methylation data at 450,000 CpGs throughout the genome, and is a sort of a quick and dirty way to measure methylation status at a whole range of sites across the genome. Looking at entorhinal cortex, as well as superior temporal gyrus and prefrontal cortex. So they took the methylation data from this array and they regressed it against the Braak stage. The Braak stage, if you recall, is a system for quantifying the rate of neurodegeneration in a given tissue or in a given sample of brain. The strongest experimental designs, I think, when you're doing this kind of thing, or really anything, is to try to convert your phenotype into a quantitative trait. If you just go Alzheimer's case versus control, sometimes your analysis, I guess, will be kind of corrupted or clouded by the fact that some people have fairly minor Alzheimer's and other people have very severe Alzheimer's. And what you'd really like is a way of quantifying the effect or the influence of disease severity on whatever you're measuring, be it genetics or be it epigenetics. So by regressing everything against the Braak stage, you have a way of doing this. And of course, these are post-mortem samples, so you can't interview the people and ask them how they're feeling. You have to look at something that's left over in their brain tissue that's fairly objective. So the authors were looking at DNA methylation throughout the brain in these tissue samples that I found, or that I mentioned, and they observed that there are two CpGs whose methylation was significantly correlated with the Braak stage in the entorhinal cortex. This is a Manhattan-style plot where, as always, the significance level is given on the y-axis and the location along the chromosome is given on the x-axis. Here we're zoomed into a place on chromosome 8, and each of these points represents a CpG that was measured by the 450K array. And you'll see two CpGs are a lot more significant or have a much more significant relationship with Braak stage than the other ones that are surrounding them. Now, if you look where these are located, you'll see, this down here is sort of a genome browser view, that they're located in the gene body of the ANK1 gene. ANK1 codes for a protein which links membrane proteins to the cytoskeleton, and it, as a gene, has been previously implicated in Alzheimer's disease. So it's always nice, I guess, when you can take a gene that has some level of connection to the trait that you're interested in, and then show, over and above that, there's also an epigenetic link that sort of is tissue specific and concerns the tissue of interest for your phenotype. And the other study that I mentioned at the beginning, De Jager et al., they found the same basic thing. There is an association between methylation at this ANK1 gene and the Alzheimer's disease progression. Now, I'm bringing this up not only to tell you that there is the potential for epigenetic contributions to Alzheimer's disease risk, and we can think of all kinds of reasons why that would happen, but to highlight an important technical consideration when evaluating these data. Now, maybe I'll just consider let me just zoom out and tell you what they're doing one more time. They're basically taking samples of brains from people with varying levels of neurodegeneration. Controls who are perfectly, at the time of death, who were perfectly healthy, all the way to Braak stage 5 or 6 or whatever the highest number is. So you're looking at people who are healthy versus people who have experienced severe neurodegeneration. You take a sample of their brain tissue, you put it in a test tube, you mash it up, you extract the DNA, and then you measure the methylation level of the DNA. And then you find that in certain cases, Alzheimer's disease brains have more or less methylation at certain genes. Now, I'm going to take a brief second to let you think of that. What could be wrong with doing it that way? What could be problematic about making conclusions from a study that was conducted using that method? So I'm just going to take a quick break and I'll let you think about that. And we're back. So, I'll just put people on the spot here. If anybody in the chat has an idea for why it might be that this is a problematic method, feel free to shout it out. There's no reason to feel bashful if you have a guess as to why I might be complaining about this type of study. And for those of you watching the playback later, maybe you're shouting at your computer screens now saying, "Oh, I wish I'd come to class. I could have had the answer to this and I would have been so favored by Dr. McKay if I had just said the answer." Yes. Okay. Excellent. So Nikita is correct. Cellular heterogeneity. That is exactly what I was listening or waiting to hear. Of course, when you have neurodegeneration, you're losing neurons. So the brains are not equal to each other. The brain of a healthy person has way more neurons than the brain of somebody at the final stage of neurodegeneration. And so if you just mash up the tissue and look at the methylation levels or the gene expression without taking that into account, you're completely missing, you could be very severely misinterpreting what's actually happening. So thank you. Thank you for that. So yeah, this is the challenge. Now look at this. This is a brain from somebody with Alzheimer's, and this is an age-matched control. So this is a healthy person on the right-hand side, and this is a person who has undergone severe neurodegeneration on the left. And there's obviously there's a difference in the brain size. The ventricles here are much larger in the AD brain, and the gray matter is much thinner. What's really happening is there's a significant reduction in both the number and density of neurons in all parts of the brain, the hippocampus and cortex in particular. So that's kind of almost a trivially well-known fact, but I'll show you this figure from this group here. Cell density is given on the y-axis. This is millions of cells per gram of tissue. The Alzheimer's brains are given in black bars, and the control brains are given in white bars. And what you'll see is that for every gram of tissue, there's a significant reduction in the number of neurons. See, neurons are much, much less in the Alzheimer's brains. And in contrast, there's a significant increase in the number of in the density rather, of other kinds of cells. Glia, you would assume, microglia and astrocytes and so on and so forth. So that in the Alzheimer's brain, believe it or not, there's actually an increase in the overall number of cells, but there's a sizable decrease in the number of neurons. And of course, neurons are the part of the brain that actually do the real work of thinking and processing information. So you have a massive loss in neurons, and naturally this means that the tissue samples are not going to be equivalent to each other because they differ so much in their cell composition. Now, how can we solve that problem? This is with a method called cell sorting. Now, Alzheimer's and neurodegenerative diseases like it are an especially important example of this, where cell composition really matters and can be a huge confound for the research. It's not the only place where it would matter, but it matters quite a bit here. there are actually computational algorithms that can estimate cell proportion. Things like CyberSort can take the expression or a methylation level at marker genes and use these to sort of infer the cell composition of the different samples. But it's all algorithmic. It's not really a real measurement, and so you can never be totally certain if it's dissociated from your variable of interest. Here, cell composition and disease state are totally confounded. People who are in the late stages of Alzheimer's are going to have way fewer neurons, and if you try to statistically adjust for that using computational algorithms, you might be adjusting away the effect of the actual thing you're trying to measure. So it's not really too practical. The only answer is to physically separate the cells before you measure them. So back in 2018, kind of in response to the work of the earlier papers, Gasparoni and company used fluorescence-assisted cell sorting, or FACS, to separate neurons and glia from 31 post-mortem occipital cortex samples from people with or without, or for people with at various stages of neurodegeneration. The paper is given here. It's in the Epigenetics and Chromatin Journal, and that's that's a much less fancy journal than Nature Neuroscience. So that shows you how much injustice there is in the world. The superior study ends up in the lower quality or the lower impact journal. But anyway, FACS was used to sort nuclei from cells on the basis of NEUN labeling. NEUN is I think I spelled immunofluorescent wrong. Okay, so ignore my bad spelling. NEUN immunofluorescent labeling. NEUN is a protein that's expressed exclusively by neurons, and thus you can label it fluorescently and figure out which cells are neurons and which cells are non-neurons. And ultimately what this lets them do is study DNA methylation in neurons and glia separately from one another. And this overcomes the major aspect or the major challenge of cellular heterogeneity that we see. They use the same 450K array to do this study, which I think is actually kind of a weakness. I mean, 450K is not good compared to whole genome methods like WGBS, but it's good enough, I guess, for these guys. And it's good enough realistically because WGBS is so expensive. So I didn't want to get into a whole big diatribe about how FACS works, although I think many of you would find it quite fascinating. So I would encourage you to look this up on your own time. What it does is almost magical. Essentially, you can feed the machine. It's just a big machine. You feed it a suspension of your cells. They could be from a cell culture, or they could be dissociated from a tissue. Whatever it is, as long as they're floating around separately in a medium of some kind. The machine will suck them up and pump them out through a nozzle. And it'll pump them out such that each cell is put into one single droplet of sorting buffer. So one droplet contains one cell. This is done through the magic of microfluidics. You can study the movement of fluids in such detail that cells can be precisely slotted into individual drops of tissue. As the cells are, as these droplets of sorting buffer, each of which contains one cell, pass through the machine, a laser scans them and scans the fluorescent signal that they express. And so, of course, this is fluorescently labeled. So you can fluorescently label things with green fluorescent protein, GFP, or you can label them with fluorescent antibodies, or whatever it is you like. But ultimately, you're trying to distinguish one cell type from another. So on the basis of this fluorescent signal, the machine can decide whether the cell passing through is a neuron or a non-neuron. And then on that basis, it will impose either a positive or negative charge on the droplet. Okay, so it's able to make this decision, tens of thousands of millions of times a second as cells fly through. It'll look at each one at a time and say, is this a neuron or is it not a neuron? Is it a neuron? Is it not a neuron? And then on the basis of that, it will charge the droplet. And then as the droplet passes through the machine, it's basically flung out the bottom of this nozzle. It passes by these deflector plates that are positively and negatively charged. And then as it passes the deflector plates, of course, opposite charges attract. So if the cell was positively charged, it'll go toward the negative plate. And if it was negatively charged, it'll go toward the positive plate. And then this imposes just enough deflection on these droplets that they travel in one direction and land in a little test tube that collects the cells. So it's a way of rapidly sorting cells using fluorescent signals. And I'll tell you right now, I'm not going to test you on this. I don't think, my kind of lame explanation is not good enough to be testable, I think. But I would say that you should appreciate, I guess, how amazing this technology really is, that you could put a tube of cells in a machine and have it sort through them at such a rapid rate. The other alternative, I guess, would be to pipette them out manually one at a time. But FACS can separate millions upon millions of cells in a few minutes and give you perfectly pure piles of neurons and glia separated from one another from the same sample of brain. So it's really quite an astonishing technology, and funny enough, it's not even really that new. Now, if you look at the methylation data from these separated neurons and glia, you'll see on the basis of hierarchical clustering that the glia and neurons separate quite cleanly. So this is a dendrogram that shows the level of similarity between these different samples. And it's kind of like a family tree, right? So the biggest difference between these samples is between cell type. So all the glia end up on this end, and all the neurons rather end up on this end. And then within each cell type, you don't see a lot of you don't see a very coherent pattern of subclustering. The control samples are the lighter colored ones, and the Alzheimer's samples are the darker colored ones. And you do tend to see among neurons, the Alzheimer's samples tend to cluster away from the control samples. And a similar tendency exists in glia, but it's not a very strong one. So it kind of tells you that genome-wide, there's actually not a huge amount of methylation difference between Alzheimer's and control brains within the same cell type. There's no clear, so within these groups, there's no clear separation between cases and controls. So that's all well and good. I don't think maybe you'd expect there would be. These neurons are dying, but it's not necessarily because they're epigenetically different. But let's go back to our question about ANK1, because this was the major finding of the previous, of the two papers from 2014 that I showed you. Now, as I said, those papers were very well done, but they could very easily have been confounded by these differences in cell composition that are inevitable as Alzheimer's progresses. The studies, I should say, show that the methylation levels increase as the Braak stage advances. So the more advanced you are in Alzheimer's disease, the higher the level of methylation is at these two CPGs around the ANK1 gene. The reality is the story is much more complicated than this. Methylation is cell type specific at these two CPGs. So in healthy young brains, neurons have higher methylation than glia, but this difference between cell types disappears with age. And this is the very kind of thing that could have contaminated the data from the papers we saw in Nature Neuroscience. Let me go over these plots. So these are the two CPGs that the Lennon et al. Paper identified, okay? And in these two scatter plots, they've got methylation level on the y-axis. It goes from 50% methylated all the way up to 100. They're as decimals, so 0.9, but 90% methylated. And then on the x-axis, you have age of the people being tested. The controls, of course, are all toward the healthier end, all toward the younger age. And the Alzheimer's patients are all toward the older age, not surprisingly. By the way, you really have to wonder, like, this is a great study and everything, but couldn't they have made these y-axes have the same scale? Like here, the top is 0.9, here it's 0.90. Why the different number of digits? You know, a little bit of attention to detail would be nice, folks. Anyway, what you'll see, also some better colors, because I find these colors very hard to distinguish. But anyway, if you look at the glia at this particular CPG, what you'll see is, if you look at the difference between neurons and glia, here are the neurons up here in this green color, and here are the glia down here. So in your 20s, the difference between neurons and glia in methylation is quite high. As you get older, the methylation level at this CPG in glia goes up, such that by the time you're old, the methylation level in glia is the same as the methylation in neurons. Now, and the same is happening at the other one too. So think about this. what is Alzheimer's disease at a cellular level? It's a loss of neurons and it's a gain of glia, various kinds of glia, I suppose. So the appearance of higher methylation isn't because methylation is changing in neurons. It's because neurons are dying off and being replaced by glia. And the glia, the older glia, have higher methylation. So it's not necessarily a change in methylation at all. It's just a change in cell composition that's being masked by this, or it's being interpreted as a change in methylation. And so the advantage of what this Gasparoni paper was able to, or of their method, is that they can show that Alzheimer's disease-associated epigenetic changes are cell type specific. So now, because you've separated the two cell populations, neurons and glia, you can run the same study, or you can ask the same question separately in both cell populations. You can ask for Alzheimer's disease-associated epigenetic changes in neurons, and you can ask for them in glia, you can ask for them separately. And it turns out that the two questions give you very different answers. So in neurons, they were able to find 954 CPGs that had significant differences in methylation between cases and controls. And in glia, I guess they found the same exact number. Or no, I guess here they're only looking at the top 1,000. So this is, yeah. So they're looking at the top 1,000 hits. And of the top 1,000 hits, half of them are unique to neurons, half of them are unique to glia, and then only 46 are shared between the two. I guess it's the top 2,000 hits. Sorry, it's still early, but anyway, the top 1,000 hits per cell type, I guess. Okay, so anyway, of the top 1,000 Alzheimer's disease associated methylation changes in neurons, 46 are shared with glia, and vice versa. So what it means is that you're to really understand what's happening epigenetically, you have to look at these two cell populations separately. And here are the methylation kind of Manhattan plots showing that showing the association between methylation level and Alzheimer's status on in neurons and glia separately. And by the way, so they did replicate this finding with this ANK1 associated CPG. So they did find the same one that the original authors found, but the signal is coming from glia, not neurons. So the conclusion here then is that as far as the epigenetics are concerned, as far as the epigenetics are concerned, glia seem to be just as important, I suppose, as neurons. Even though Alzheimer's is seen typically as a neurodegenerative disease, it would point to some importance for the glia as well. And there's a great deal more you could get into if you wanted to pursue that further. But I think it's enough to say that you can't. You have to be very aware of cellular heterogeneity. Of course, doing single cell sequencing would be the next step above this. Then you can get into various subtypes of neurons and subtypes of glia, and perhaps you'd be able to find which particular subtypes were the ones that were really experiencing the disease-associated changes. But that's a question for another day. Anyways, my friends, that's actually all I had for you today. I guess I kind of got through it a little bit quicker than I was planning, but I don't think anyone's going to complain that today's lecture is one of the shorter ones. Now that we've kind of gone over a lot of the basic lessons about genetics and epigenetics that I wanted to teach you in the previous lectures, now it's really just a matter of going through some of the data highlights to help you understand each condition. So that will mean the lectures theoretically could be quicker from here to the end. So hopefully that'll be good for everybody. Anyways, friends, we'll shut her down there for today. Hopefully on Wednesday's class, I'll be able to go over the results of the midterm with y'all once the grading's done, and discuss the housekeeping matters for the last little bit of this course. And I know that you're all excited about American Thanksgiving coming up this week, and Black Friday and all the rest of it. So enjoy. I guess you don't get the day off, but enjoy yourselves nevertheless, and we'll see you on Wednesday. Thanks everyone for listening.